Hello all, this is a quick video as an introduction to genetics. Our lesson objectives for today are as follows. To review some key vocabulary relating to genetics, which you might have seen previously. To understand the role and importance of genes. And to define and consider an example of DNA mutation. The essential idea overlying topic 3 is that every living organism inherits a blueprint for life from its parents. So firstly, what is a gene? According to the understanding, the definition is a gene is a heritable factor that consists of a length of DNA and influences a specific characteristic. So the key idea is that it can be inherited. It can be passed from one parent to the offspring. It consists of a length of DNA. So DNA is actually the chemical which makes up a gene. And as we will see, a gene is simply a small part of a chromosome. And a gene determines a specific trait or characteristic. A gene also occupies a specific position on a chromosome, which we will see in a second. And this position it occupies, which is always the same, is called its locus. So already we can see there's some new vocabulary building up here. There are roughly 21,000 genes in humans, and they are organized, as you may know already, into 23 pairs of chromosomes located in the nucleus of our body cells. One copy of these pairs of chromosomes comes from the mother, and one comes from the father. And this is sexual reproduction, and this pairing happens during fertilization. Here we can see a karyotype of human beings, which you might have seen before. So we can see here at the top our 22 pairs of autosomes. So these are our non-sex chromosomes. So we have 22 pairs of those of differing sizes, and you can see those alternating sort of black and grey bands representing the different sections where genes might be located. And down the bottom right-hand corner, we can see one pair of sex chromosomes. In this case, indicating a male, as it has an X and Y chromosome. And of course, we get our individual members of those pairs from the sperm, which is bringing 23 chromosomes, fertilizing the egg, which also has 23 chromosomes. And that's where we get our diploid number of 46 chromosomes. So one of the challenges of this topic is the new vocabulary that you need to learn. So please stay organized. There is no more important unit for building up a glossary than this unit right now. So we've seen gene. We will see allele, chromosome, DNA, autosomes, and also sex chromosomes. So please make sure in your mind you have a really good firm definition and understand clearly what these terms mean. This is a diagram from the book which helps to put everything in context for us. Now it doesn't include the term allele, which we'll deal with in a second, but you can see there on the far left-hand side the cell with a nucleus which contains in a human being, this would be the 46 chromosomes. 23 pairs, so we can see our chromosome pair there, and then we can see even further, looking into the chromosome itself, that DNA is the chemical which makes up chromosomes and therefore genes. We've seen this previously, a very simplified version of DNA, and we'll look at this in a lot more detail later, but our DNA is made up like a twisted ladder, this double helix classic shape, and we have 4 bases which are paired up inside of that DNA, if you remember. Thymine pairs with adenine and guanine pairs with cytosine, and this actually makes up that blueprint. So this is the genetic code, the order of these bases. So what do genes actually do? Now, genes are so important because they influence us, because each gene contains the code for synthesizing a specific protein. So protein is the key word here. Proteins, as you know, have many different roles in living organisms. So they might form the structural part of our muscles or bones or our connective tissue. They might be heavily involved in the immune system, keeping us healthy. And really, really importantly, they are also enzymes. Now, as you know, enzymes are critical for the biochemical reactions, the metabolism in our body. So essentially, genes make or contain the code for proteins, and proteins determine so much of what we look like, how we act, and how we function biochemically. Basically, genes determine who we are and how we function. We haven't looked at transcription and translation, but this is a very simple diagram. We'll look at those two processes later. But we can see here a gene, through transcription, it leads to RNA, and then through translation, the RNA gets converted into our protein. So the gene contains the original code, leading to the single protein being produced. And as we can see down the bottom there, this can be influenced by environment or lifestyle or disease. So there is this interaction between the genetic components, which is our genotype, and the environment around us, the phenotype, which we probably reviewed previously. I will get you at some stage just to watch this very short video. It goes for about 2 minutes, and it's a nice video, very simple, puts everything into perspective. So please, if you feel it's the right moment, pause the video and watch this quickly. Just a really quick review of sexual reproduction. So we can see here, this is the human example. So we can see down the bottom left-hand corner there is our male and female with our testes and ovaries producing our sex cells through the process of meiosis, which we will look at soon. So these cells are produced in humans. The ovum or the egg cell will have 23 chromosomes. The sperm cell will also have 23 chromosomes, represented there by a small n, representing that this is the haploid side of the sexual reproduction process. Then we have the joining of these two cells in fertilization. And once fertilization occurs, we have a diploid zygote represented by 2n, containing 46 chromosomes. That single cell will then divide by mitosis, and development will occur, and eventually we will have the birth of a small human child that has gone through the process of sexual reproduction. We see similar process in plants. So if we start with a flower, which is in the top left-hand corner, we can see the stamen there and the anther, which is the male part of the plant. It contains the pollen grains, which would be haploid at this stage, containing half the genetic information for this plant. And through the process of pollination, whether carried by the wind, water, or a bee or another insect, the pollen grains there will attach to the stigma of the flower, the female part of the flower. A pollen tube will grow down, it's represented by that yellow line, and the sperm cell will come from the pollen and fertilize the egg cell, which is contained within the body of the flower there. Fertilization occurs, we have the seed development, which is now diploid, containing the full complement of chromosomes for that particular plant, and then the seed germinates to produce a plant which then matures into the flower that we can see. So we can see there a similar process to what happened in humans. So we will review both of these in more detail later, but just at the moment, have a good look at that and trying to keep all of this in mind as we move forward. Okay, this is where we get to a new word, allele. So according to this understanding, the various specific forms of genes are called alleles. So the gene codes for a particular trait. The classic one we use is always eye color. The allele would be brown eyes or blue eyes or green eyes. So the particular type of or the specific form of that gene is called an allele. It's really important, guys, that you move forward having a solid understanding of these two terms. We can see here a nice representation. So we have a pair of homologous chromosomes. At the very top there, that green band represents, let's say, a gene on the female chromosome on the left and the male chromosome on the right. In this case, the gene is the same. So if this was eye color, again, a classic, but much more complicated in reality, this might represent blue eyes and blue eyes. As we move down, we can see there's some genes at the bottom in the same locus, the same location, but in this case we have different versions of the same genes. So at gene 2, we have the red band on one side and the green band on the other. This represents different alleles of this gene. We're going to provide one mutation example during this video. We'll look at lots more in class. And this one is a protein called transducin. Now, transducin is really important in transmitting information from the eye to the brain and allowing color vision. It's a protein. As we know, proteins are determined by the genes. It is located, or its locus, is on chromosome number 1. A mutation, or a change in the genetic code, leads to complete achromatopsia, a rare genetic disorder. Say that 3 times fast. This mutation involves a single base substitution where the C is replaced with a T at position 235 of the gene. So it's a single base which has been switched. Color vision is therefore a genetic trait. Okay, it's linked to the genes. And what that essentially means, here is our transducin, a representation of it. And this is the transducin gene. So it's located on number 1. And we can see the sequence there starting at position 229. And if we go through, we have CACCAGC. And this is the correct genetic code. It will lead to transducin protein, and color vision is possible. Down the bottom, however, we can see that that C with the underline has been replaced with a T. This is the mutation, a changing of the genetic code. The protein is not formed correctly and color vision is not possible. And what we essentially end up with is something like this. So if you don't get the right transfer of information from eye to brain, you would be seeing on the left-hand side of the picture there, as opposed to the wonderful dawn scene on the right. And this understanding sort of targets that and states that alleles differ from each other by one or only a few base pairs only. So you can see there that these different forms of the gene arise from very small changes or relatively small changes. We will look at the sources of mutations and other types of mutations a little bit later. And mutation leads to new alleles. Now these might be adverse alleles that cause us problems, or they could actually be beneficial alleles, and they might be important in natural selection. So again, new alleles are formed by mutations. Mutations, random, rare changes in the genetic material. We'll look at the sources of these later, the possible sources. Mutations may be negative, so transducin, I think you would agree, is a negative trait, and we'll also look at cystic fibrosis as an example. Mutations might actually be positive. Okay, one example we'll look at, which you might argue for, is sickle cell anemia in places where malaria is prevalent, and also this one here, the LRP5 gene, which actually helps the immune system make a certain surface protein. And HIV recognizes this protein. If there is a mutation in the LRP5 gene, that surface protein is not produced, and this person essentially has HIV immunity because the HIV virus cannot recognize that protein. So there's an example of a rare mutation which can actually benefit someone. And also evolution by natural selection. The driving force for natural selection is, of course, variation within the population. Variation comes from genetic variation, and this genetic variation is essentially multiple alleles for a particular genes. Those alleles arise by mutation. So we can see here a simplified diagram of how mutations can lead to, down the bottom row there, variation in a population. And this can eventually lead to evolution by means of natural selection. In new alleles, when a mutation is successfully passed from one generation to the next, it becomes a new allele. It is a new version of the original gene. Okay, a little bit of homework before next class, please. I just simply want you to read and take notes from pages 119 to 122. This is from topic 3. This is essentially a review of what we've covered together today, with a little bit of information just to fill out your knowledge. So please walk in with a very good understanding of those terms, and we'll start to consider some specific mutations and cases. Thank you very much.